I'm an associate professor at the University of Auckland. I've got a PhD in vaccinology.
So is mRNA vaccination a form of genetic modification?
No, the product itself has been developed using genetic engineering, but the product itself consists of RNA. Now, RNA cannot modify your genome, because in order to do that, it would, first of all, it would need some additional specific instructions, but it would also need to gain access. Once it's inside your cell, it would need to gain access into the nucleus where your genome is, and it can't do that. So no, it can't.
Why can't it infiltrate into the nucleus?
It needs extra bits and extra instructions, which it doesn't have. It's a one-way trip. You can get messenger RNA comes out of the nucleus, but it doesn't go back in, and it also disintegrates quite quickly.
My name is Phillip Buckhaults. I have a PhD in biochemistry and molecular biology. I'm a cancer gene jock. Basically, I do cancer genomics research at the University of South Carolina.
But the product itself consists of RNA. Now, RNA cannot modify your genome.
The Pfizer vaccine is contaminated with plasmid DNA. It's not just mRNA. It's got bits of DNA in it. This DNA can and likely will integrate into the genomic DNA of cells that got transfected with the vaccine mix. This is just the way it works. We do this in the lab all the time. We take pieces of DNA, we mix them up with a lipid complex like the Pfizer vaccine is in. We pour it onto cells, and a lot of it gets into the cells, and a lot of it gets into the DNA of those cells, and it becomes a permanent fixture of the cell. It's not just a temporary thing. It is in that cell and all of its progeny from now on forevermore. Amen. So that's why I'm kind of alarmed about this DNA being in the vaccine. It's different from RNA because it can be permanent.
I do have a long history in this field, and I don't have a history that's really financially tied to this narrative, so I can call balls and strikes pretty fairly. I've had about a 30-year experience in this field. I started off my career at the Human Genome Project at MIT. In fact, that robotic pipeline you see up there in the corner is something that our team engineered for the Human Genome Project to purify plasmids. So we live, ate, and breathe plasmid DNA purification on the Human Genome Project.
But the product itself consists of RNA. Now, RNA cannot modify your genome, because in order to do that, it would, first of all, it would need some additional specific instructions, but it would also need to gain access. Once it's inside your cell, it would need to gain access into the nucleus, and it can't do that. So
when you hear a lot of the responses to this work, they'll say, well, it doesn't matter, it's never going to get to the nucleus. There's two things wrong with that statement. One, there's a nuclear targeting sequence in there that guarantees it will get to the nucleus. Second, these LNPs go to cells that are dividing, and when cells divide, there is no nuclear envelope protecting the DNA. They have to go through cell division, and that dissolves during that process. So when they're talking about this not getting to the nucleus, these people are doing a smokescreen on you. They're not being honest with themselves that cells divide, and when cells divide, there is no protection of this DNA. So that's something to watch out for. When you see people saying it doesn't get to the nucleus,
it would need to gain access into the nucleus, and it can't do that.
That's a sign that they are trying to lie to you.